Hello, everyone. Today we're going to be talking about how to take your 16S sequences into biological information and data through analysis. And so we're just going to focus right now, our goal today, on getting to a feature count or contingency table. But this is kind of an overview of what the process looks like of handling 16S data. So we start off with our DNA, which gets sequenced and then converted into some kind of signal. With Illumina, it'll be fluorescent signal, or with Nanopore, it'll be a electrical signal. This will get base called and then turn into a Fastq file. And then we're going to do some bioinformatic magic that we'll talk about today, eventually getting interpretable data that we can actually help us understand what's happening in the microbiome environment. I wanted to give a really big or brief recap on the research cycle in microbiome research. And so we start out always with having a research question, and we want to make sure this is a clear and testable hypothesis. Here's just one example of such a hypothesis. So an 8-week course of drug A will reduce butyrate producing bacteria. That's a clear and testable hypothesis. Now, this research question, whatever it is, can then drive, okay, our model and what samples we want to collect. Let's say we can collect mouse samples, stool samples from mice, we can collect oral samples, we can collect human vaginal samples, we can collect soil samples, whatever the question might be. And so one thing we want to keep in mind is to make sure that everything is sterile as we're collecting it, because we don't want to introduce microbes in our sample that weren't there originally, right? So that's really important. And then we want to store immediately into -80°C freezers and avoid taking them in and out of the freezer, because that will degrade DNA faster than the microbes. And then we want to make sure that we include appropriate controls, whatever that might look like for your specific experiment. Document metadata, so as much information as you can: sex, weight, age, even metadata that you might not think is important at the beginning. It might end up driving some differences in the microbiome later as you analyze. And then making sure you have enough samples to work with. And so we want to make sure we have enough statistical power with our sample size. And then we're off to extract the DNA from the microbes in our sample. Okay, so we want to tailor the DNA extraction method to the sample type. So there are specific kits for soil and specific kits for human stool, things like that, that are available. Things to keep in mind are the amount of biomass you'll find in your sample, or if there's any potential inhibitors that might prevent, let's say, PCR down the line. There are kits that are able to handle these situations better. Low biomass you might find in sometimes like plaque samples, or they're really hard to extract, or in a situation I've been in where I had environmental samples from like the Arctic that had very, very little microbes in it. So those are things to keep in mind, but stool, for example, will have extremely high biomass. And there are different mechanisms for breaking open the bacteria. We can either go through like the bead beating mechanisms, which will use mechanical forces, or a chemical lysis, because we ultimately want those bacteria to break. Some are more stubborn than others. You'll have to optimize your protocols. Include technical replicates if necessary. And then also keep in mind the batch effect. So monitor the batch effect, which means if you're performing DNA extraction in batches or sequencing in batches, the small variations that you impose can create variations down the line that were not biologically truly true variations from your sample, but rather that were introduced just by these different batches that you had. All right. And then we also need to select appropriate variable regions. Okay, so with our 16S gene, which I'll talk about in a moment, you want to make sure you pick the right primers, the right regions, to answer the questions that you're interested in. So you might pick a different set of primers if you want more resolution, more specificity of the microbes of interest. And then also minimize the cycle number to reduce noise and bias. So we don't want the PCR to cycle more than absolutely necessary. Using a high fidelity polymerase will prevent any errors being introduced in this PCR process. We definitely don't want errors to keep carrying down, right? And then also using a no template control. This is super important because as we're going through and performing PCR, there's always a risk of accidentally contaminating our samples. And so that means maybe a sample that had a lot of DNA in, you added in a sample that shouldn't have had any DNA in it, and now it's showing up as if it worked. However, if you used a no template control, this will allow you to track any contamination that you might have had, because you're going to be carrying, let's say, the same like master mix or whatever it is throughout the samples, depending on how you like to prepare your PCR mixture. Okay. Now, once we get to the sequencing step, so library preparation and sequencing, which is just preparing the pool of amplicons or PCR products from all your samples, and then sequencing, we want to account for run variation, again, which that batch effect. Ideally, we'd like to put all of our samples in one batch, and then determine the appropriate depth. So if you want to really find like rare taxa, you might sequence deeper and make sure that you have many, many, many reads. But if you're just trying to get like a rough overview of what's going on in the microbiome, you might not need as much sequencing depth. Okay, and then there's the question of paired versus single end reads. So paired end takes into account both ends and looks at the forward and reverse sequences. But overall, paired is nice because it'll allow you to kind of double check your sequences. And then platform selection will depend on, again, the kind of sequencing that you want to run. So they all have their strengths and weaknesses, right? The nanopore and Illumina and PacBio all have their strengths and weaknesses. Okay, so there's also data analysis now. And then kind of an overview is that we want to pre-process, turning those signals into sequences and cleaning up those sequences, basically assigning taxa. So assigning, okay, in this sequencing read, this means it's that microbe or that type of microbe, right? And then creating a feature table. That's where we're going to end today, but then it keeps going, where we create a fileseek object, and then look at the actual diversity, abundance changes, and statistics in regards to our data. So that's just a little overview, so you're well acquainted. And then now let's give a little recap on the 16S gene. The 16S gene, again, is a part of the 30S subunit of the ribosome, and this is found in prokaryotes, archaea. And we're going to see one copy per genome, most likely, but there are cases where there are more than one copy per genome. This will allow us to get more or less a proportional representation of how many of each microbe there were. However, we're not really going to get absolute counts. In the end, when we analyze our data, what we're really analyzing is not absolutely how many microbes there were in our sample. We're going to be looking at more of a proportion or composition, right? Especially because we amplified with PCR. The total length here that we see is 1.5 kb. Right, so that'll be over here, 1.5 kb, kilobases long. And if you want to sequence the entire thing, you can do that with nanopore. However, most people just look at this portion right here. And so these peaks and troughs basically represent how conserved or variable the sequence sections are. And so here is a very conserved region, as well as here here. And so those are really great places to put primers, actually. But here, this variable regions, because we also want to cover the variable regions, those are what's going to tell you who's who. That's what really differentiates between the microbes is these variable regions. However, even if you just take this part right here, you're still not going to get like a species level resolution. To really understand which species are there, you have to be really specific, which means taking into account the entire length of the 16S gene. Okay. And also just to emphasize that we are targeting DNA, not the RNA, despite it being used as an rRNA, right? Here's the example. Now we're off to amplifying our sequence. So we want to make sure we design our amplicon appropriately. These are just some of the basic components that most amplicons will have. We have our target region. This is going to be whatever you decide, depending on your primers, which regions you decide to use in a 16S gene. And then, yeah, so those will your respective primers. And then we have something called barcodes over here, or also called like an index. Those are there because you're eventually going to pool all your samples into one tube, okay? And then you'll have to re-identify them later. And so having these barcodes basically tells you which samples each amplicon came from. So every sample will have its own barcode, which is just basically a small sequence, a small chain of nucleotides. And then we have here also adapters, and these are going to be different for every single library prep kit, and those will be specific to the sequencer. And however they work, they're meant to facilitate sequencing later down the line. Here's also an example. Here they're called index instead of barcode. But again, they have those primers. This is looking at the variable 4 region, roughly 254 base pairs right there. And we see this is just an example of what multiplexing and demultiplexing will look like. So that's, let me show you here. Basically, we're combining all of our samples into one tube in an equimolar pool. So that means that each sample is going to have an equal concentration, so they have equal representation. And then we're going to demultiplex them later bioinformatically. And what we find is that we're able to tease apart which sample has which reads, and then they'll have their own folders where all those reads go respective to their sample. This is kind of a visualization of Illumina sequencing by synthesis. So this is used of a fluorescent signal. And so as the DNA is polymerizing, it'll have a fluorescent nucleotide that will come in one by one and then be read as a fluorescent signal. This will get base called into a Fastq file and then pre-processed, demultiplexed, as I just explained, trimmed and filtered, so all cleaned up. Let's take a look at that. Okay, here is an example of that quality score or that Q score throughout the sequence. You'll see it says cycle here at the bottom. That's referring to which nucleotide we're at. So here at 250, that's the 250th nucleotide. And this is normal where we see the quality will decrease significantly. This is normal. This is due to many factors, but just less efficient sequencing is expected as time goes on. We have a cutoff here of 10, which is pretty low, right? And with nanopore, we can expect much lower than Illumina. This is Illumina right here, very nice. We can actually kind of get rid of this portion because we have a paired read, right? So this is the forward. I believe this is the forward strand. Let's say this is the forward strand on the left and the reverse strand on the right. So we can cut this off and still have our full sequence. However, we need to keep something in mind. which is we need our strands to still be aligned with each other, right? Because let me show you what happens. So let's trim that portion off, and here we're going to be trimming the end of this one. We want to make sure there's still enough overlap that they can be matched back together. One way to do the math for this, okay, I've accidentally written over that. So an easy way here is if you're trimmed forward strands, so let's say it was originally 250, and now we trimmed it down to 200. We'll do the same with the reverse. But then originally, like I said, it was 250. And so the overlap here is going to be 150. That is pretty good, right? We want something over 100 ideally would be good. Okay. And then we will cluster the sequences in either two different methods, OTU or ASV, and then assign taxa so that we can identify, okay, this read means this microbe. And we're going to end here with our table. Okay, just a quick recap. This is like the phylogenetic, the taxonomic order of things, right? And so we go from phylum all the way down to species, but with short reads, we're really only going to get down to that genus level, which is not bad if that's what you want. So OTUs are operational taxonomical units, and this was developed quite a while ago, but basically it clusters sequences at defined similarity thresholds. Let's say 97% is a common threshold that people use. And this is kind of what it looks like. So these aren't perfectly, the same color, but they're close. They're pretty close. So like, let's say 97% close. And this means their sequences have 97% similarity, right? And so this was done especially during early 16S sequencing, and this is mostly because many of the sequences were shorter, so with less resolution. They had higher errors and weaker computers, and so they needed ways to kind of group things together and make it easier, right? And so OTUs were able to, in this case, reduce noise and then make samples more comparable. There are two kind of types of OTU grouping. And so there's de novo, which doesn't rely on reference, and then there's closed reference, which does. What does this mean? De novo basically is grouping without a database. And so this is great if you have like unknown taxa. However, things get messy if you want to add new samples, introducing new sequences, because then you'll have to rearrange all of the OTUs again, right? And it's not very reproducible because, again, it's so dependent individually. However, if we have a closed reference, it will compare every read to something in a database, and if it doesn't recognize it, it will throw it away. And so, yes, only keeping OTUs using reference database. This is much more reproducible. However, it is very biased depending on which database you use, right? Okay. Then we have ASVs, which are amplicon sequence variants. Okay, this is very different because it is much more specific. It basically groups now every unique sequence with even a variation of one single nucleotide in its unique groups. This is highly more specific, with much more resolution and precision. And then this is like an example of what that might look like. So you would have like anybody of the same color exactly would be its own ASV. In this case, ASV1, ASV2, ASV3. This does not rely on a reference. However, you might imagine that, sequencing is not error-free. There are going to be typos. So how do we make sure that it's not creating a new taxa, a new genus, a new species, just because it found a typo somewhere? Well, it takes care of this with different methods called like denoising. And one technique that it uses out of a couple is, for example, if it finds something that's very rare, maybe that just happened once, it might discard that and not consider it an ASV. And then it also removes chimeras, which are kind of accidental combinations of amplicons during PCR. I do want to highlight that this is becoming now the standard expectation in microbiome research, just so you guys know. Once we have the groupings, OTU or ASV, then it's time to align and say which OTU is what bacteria, which ASV is which bacteria. In this case, let's say it's identified OTU1 as Roseburia, OTU2 as Klebsiella, and so on. Commonly used databases are like Silva or the GT database. And these are probabilistic alignments, right? This is kind of what your contingency table is going to look like. So you'll have, oh, by the way, this is called not just contingency table, but also ASV table, OTU table, feature count table. And so you'll have your samples on the left, and then ASV1, ASV2, ASV3, and you'll see how many of each ASV was found in each sample. I wanted to highlight here the 0 here, which is very normal in microbiome data and is very problematic as well, just for statistic purposes. And so that's something we need to deal with. I just wanted to highlight that. That's something we're going to deal with later. And then I also wanted to just say one more time about how big of a problem misrepresentation can be with microbiome data and how to prevent that. So we start off with our actual sample and the different species that are there. We perform PCR. And now we've already, in a way, misrepresented who is there. Because certain species are going to have 16S genes with higher affinity in their primers than others, for their primers than others. And so there's going to be a primer bias. Some amplicons are going to amplify more than others, right? And this is also made worse by cycle number. If you increase this too many cycles, it's going to get even more disproportional. There could be a lysis bias. So some microbes might be much harder to lyse in the beginning than others. There might be more than one 16S copy in their genome. And then also PCR can generate some of those chimeras, right? Okay, then you're off to sequence, and that can also introduce some errors, sequencing errors, and then also when you're pooling, you want to make sure everything has been pooled equally. We want every sample to be represented with the same DNA concentration of their PCR product, and you end up with ASVs, right? This is kind of what that looks like when you misrepresent a sample using variable concentrations of DNA. So let's say you have sample A and sample B, and the concentration of DNA for sample A is 100, and for B it's 50. Well, if you go into PCR, you're likely going to get way more of sample A, and so it might represent the diversity much higher in that case. But that's not necessarily what was really in the sample. It's just that you extracted more DNA in the beginning. So you can normalize that by putting into your PCR mix the same amount of DNA. Okay, so let's say you do that, and then in your PCR, just because of variation and randomness and primer bias and all these things, you still end up with different concentrations of your PCR product. So you might end up with 25 in sample A and 60 in sample B. You need to normalize this again before you move forward with sequencing. At this step, I might also mention, it's important to also clean, okay? So what do I mean by that? Well, this will be specific to your kits. However, when you're performing PCR, you're adding polymerase, your protein, you're adding different ions and things like that. So you want to clean that up using DNA purification method. Like, so for example, DNA binding beads is a good way to make sure you clean that up. But then, yes, you want to normalize. And so all of the samples are being represented equally as they're going into that equimolar pool to be sent off for sequencing or to perform sequencing. All right, so that's all for right now. But if you guys have any questions, let us know. And next thing you'll be doing is hands-on. So we'll be here with you the whole way. Take care. Thank you.